Yes, we can see it.
Thank you so much. Welcome to tonight's session on Mendelian Inheritance. We're happy to have our expert tutor, Mr. Adisu Timilei, with us to dive deep into this topic. Let us get ready and get engaged. Welcome, sir, Mr. Adisu Timilei.
Yeah, thank you, ma'am.
Please write down.
Okay.
I said, please write down. Can you continue, sir?
Good evening to my world intellectual family. I welcome you all to today's presentation on Mendelian inheritance.
Sorry, sir. Maybe you should stop sharing your screen.
Okay, yeah, sorry. Good evening to my world intellectual family. I welcome you all to today's presentation on Mendelian inheritance, segregation to Punnett square. Today we explore the fundamental principles of genetics discovered by Gregor Mendel. We delve into the concept of segregation, how alleles separate during gamete formation, and how Punnett square helps us predict genotype and phenotype probability. So in this presentation, we cover Mendel's law of inheritance. We will also cover the concept of segregation and independent assortment, how to create and interpret Punnett square, applications of Mendelian inheritance in genetics and real-world scenario. So by the end of this presentation, I believe you will all have a solid understanding of Mendelian inheritance and how it forms the basis of modern genetics. So let's get started. So introduction. Mendel's laws of inheritance. The law of inheritance was discovered by Gregor Mendel in the 19th century. He is the father of genetics. So this law revolutionized our understanding of how traits are passed down through generation. Mendel's law is the study of how traits are passed down from parents to offspring. So that is what Mendel's law is the study of how traits are passed down from parents to offspring. So the importance of Mendel's law of inheritance is to understand Mendelian inheritance helps us predict the probability of certain traits and diseases. So Mendel's laws of inheritance. Mendel's law describe how genetics traits are passed down from parents to offspring. So it's the law that describes how genetic traits are passed down from parents to offspring, either in plants or in man. So it's the law that describes how the genetic traits are passed down from parents to offspring, and the law includes the law of segregation and two the law of independent assortment. The law of segregation and the law of independent assortment. So let's take it one by one. So let's start with the law of segregation. Law of segregation. The word segregation also means separation. So wherever you see segregation, it also means separation. So the law of separation. Each pair of alleles separates during gamete formation. Each pair's alleles, that is different form of gene, separate during gamete formation, resulting in each gamete receiving only one allele from each pairs. So in other words, when an individual produce gametes, that is sperm or egg cells, the alleles for each gene separate and each gamete receive only one allele. This means that each gametes, we have a unique combination of alleles resulting in genetic variation. So the example of law of segregation is a parent with genotype BB can pass either B or B allele to offspring. That is, will be the dominant and the recessive. So a parent with genotype B and B, that is maybe the father is the dominant and the mother is the recessive. So they will pass either B or B allele to offspring. So that is the example of law of segregation. So as we can see in the diagram, as we can see, the blue, this is the blue, that blue is the capital B allele, while that red is the small letter B allele. So when it was passed to the offspring, as we can see, so it was separated, the alley was separated, both blue and red alley, they were separated. So it finally result to the it's okay, yes, the alley was segregated into separate daughters cells or gametes. So it resulted into separate daughter cells or gametes. So let's quickly go to the law of independent assortment. The law of independent assortment. The term assortment means distribution or grouping. The word assortment. So anywhere you see the word assortment, it also means distribution or grouping. So this explains how alleles for different genes are sorted independently during gamete formation. So this explains how alleles for different genes are sorted independently during gametes. So that means they are grouped. I mean, they are grouped. They are distributed during gamete formation. So this law allows us to predict the probability of different combinations of alleles being passed to offspring. So the example of independent of the law of independent assortment is a parent with genotype BB and genotype RR can pass combinations like BR, BR. Okay, now let me to explain this. A parent with genotype capital letter B and small letter b, and also a parent with capital letter R and small letter r can pass combinations like capital letter B and capital letter R, and also can pass combination of capital letter B and small letter r, and can also pass combination of small letter B and capital letter R, or small letter B and R and small letter R. So as we can see in this diagram, in the first diagram of law of independent assortment. So though it's not really clear, but I would like to go to the Punnett square so for better understanding. So here in the diagram we have that genotype RY. Okay, both parents are genotype R and Y. So that is two seed. They has two shape seed. So now one of the shape seed is R and Y, and the other is R and Y. So now in our Punnett square, so the one in the right hand, that is RY, and the one at the top, that's same RY. So now when
Excuse me,
the
sir.
combination.
Excuse me, sir. Am I in the
Oh,
right place or should I go back?
what is it?
Should I go back or I should stay here? Punnett Square.
Okay, yeah, stay there.
Can you see my screen?
I'm coming, please.
Okay. Thank you.
Okay.
Should I go back?
No, I can't see your screen.
Okay, it's fine. Can any other person see my screen?
Yeah.
Yes, I can see your screen.
All right, sir. Thank you.
Okay. Yeah, can I continue?
Yes, sir. Please.
Okay, all right, ma. Yeah, yeah, I'm sorry.
Yes. Sir.
Okay, so I want to discuss uh okay on the diagram on law of independent assortment. So I'm explaining the diagram of law of independent assortment.
Assessment. Yeah.
So now, the combination of both parent genotype. The first combination will be, you know, it's just like I think in mathematics when we were discussing about graphical in simultaneous equation. So this ry, so they want to share the combination, it will be ry, okay, two capital letter r and two capital letter y, as you can see it here, two capital letter r and two capital letter y. The other one is two capital letter r, one capital letter y and small capital letter, and one small letter y. So the third one will be one capital letter r, one small letter r, and okay, one capital letter y and small letter y. So the fourth one will be, the fourth combination will be one capital letter y, capital letter r,
R. Yeah.
small capital letter, one small letter r, one capital letter y and small capital
Small letter
and one
Y.
small letter y. I'm sorry. So the second combination, yeah, the small combination will be one capital letter r, one small letter y. So the combination to the other parent will be two capital letter r, one small letter y, and one small letter y. That is two capital letter r, one capital letter y, and one small letter y. So the second row it will be two capital letter r and two small letter y. The third column will be, one capital letter r, small one small letter r, and one capital letter y and small letter y. So the fourth column will be one capital letter r, one small letter r, and two small letter y. So okay, I just keep it. So now the probability of the red flower, the probability of the red flower will be 9. As you can see, the red, the one that is designated with red. So it has, let's read it together. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So it will be 9 flowers, 3 blue flowers, 3 green flowers, and 1 yellow flowers. So that is the probability. So it will be 9, 3, 3, 1. So that is the example. That is the explanation for the first diagram. Why the second diagram on that same law of independent assortment? So independent assortment occurs during metaphase 1. That is, it creates genetics variation between gametes. Homologous pairs of chromosome lines up in the middle of the cell in a random order each time. So that is, during meiosis 1, tetrads can line up two different ways before the homologous separation. So before the homologous that is of the same of the same parent, so they have the same genotype. So that is homologous. White heterologous means different genotype. So as example is the one we did earlier. So now let's go to the expression to the diagram. As you can see, okay, we want to discuss about the eyes color and the hair color. So we have one of the parents has brown eyes and black hair, while the other parent has blue eyes and red hair. So now when we want to line it up randomly, so as you can see, this the first one is for one of the parents, and the other one is the for the, maybe we should use the first one for the father while the second one for the mother for better understanding on it. So the there will be one there is homologous. The homologous gene is in pairs. The chromosome is in pairs. So now, so there's probability that the offspring will have brown eyes, one will have brown eyes and black hair, and the other one will have blue eyes and red hair in both okay on both parents. So they may have one brown eye, brown eyes and black hair, and the other also will have blue eyes and red hair. So the other one also might have brown eyes or red hair, or blue eyes and black hair. So that will be the probability. So the probability of the offspring to have brown eyes and black hair is one, and the other one is to have blue eyes and red hair. So that will be 2:2. It will be 2:2 because they are the chromosome, the homologous chromosome are in pairs. So let's go to the next slide. So we want to discuss on the Punnett squares. That is the next the next slide, Punnett square. So what is Punnett square? Punnett squares are a graphical representation of the possible genotypes and phenotypes that can result from a cross between two parents. A Punnett square is a square diagram that shows all possible combinations of alleles that can result from a cross between two parents. So that is Punnett square. That is how Punnett square is being represented. So it's a graphical representation of the possible genotypes and phenotypes that can result from a cross between two parents. So how is Punnett square create? How do we create Punnett square? So one, we have to determine the genotypes of the two parents. Though we will still go to some examples, so but let's know this first. So we have to determine the genotype of the two parents. Two, we have to identify the alleles involved in the cross. Three, we have to create a square with the alleles from one parent on the top and the alleles from the other parent on the side. Four, we have to fill in the possible genotypes by combining the alleles from each parent. So this is how to create Punnett squares. So now let's look at two examples, but okay, yeah, let's look at the example and how Punnett square is being applied. So example one, as we can see in the diagram, one parent is at one side and the other parent, that is mother, is at one side while father is on top of the Punnett square. So the mother has is black in complexion, and the mother has brown hair, while the father also is black in complexion, and also the father has brown hair. So the probability will be, so we are going to take, okay, the probability that the offspring to be black in complexion is higher than the probability of brown in complexion. So that is the probability that the offspring will be black. As you can see now, when the dominant and the receiver now, so both mother and father, they are both of them are dominant, so that is why it is the same black. So in the other column, both the mother and the mother is the dominant, while the father is the recessive, I mean. So in the second one, concern the hair, the father is the dominant, while the mother is the recessor. While in the other here, that is the other prediction. So the mother, both the father and the mother is the recessor. So now in the our results of the prediction will be, we have two capital letter B in the first column. In the second column, we have one capital letter B and one small letter b. In the other row, we have in the first column we have small letter b, we have one small letter b and one capital letter b. In the other column, we have two two small letter b. So that is, okay, so that is the probability for black is 75. As you can see, we have three, that is 25, 25, 25. The whole square is 100. The whole square is 100. So we have three blacks and one brown probability. So we have three brown, that is 25 times 3, that will be 75.
Yes, sir.
And the other one brown will be, okay,
25.
can I go on?
Yes, sir.
Okay. Yeah, the other one is 25. So the probability of, so the probability of the offspring to be black in complexion is higher than the probability of brown in complexion, as you can see. So the probability that the offspring will be black in complexion is higher than the probability that the offspring will be brown in complexion.
Completion.
So that is example one. So let's look at another example of Punnett square. This is another example. Now we want to deal with the eyes color. So now, as you can see, in one side we have two capital letter B, and in the on top of the square we have two small letter b. So that is, the two capital letter B stands for brown eyes, while the two small letter b stands for blue eyes. So now, when there is okay, when there is one alley of capital letter B and one alley of small letter b, so it will be, the result will be bb. So when there is one alley of capital letter B and small alley of small letter b, it will also be bb. So all the possibility will be capital letter B and small letter b, that is dominant B and recessor B. So the probability is that all the offspring will have brown eyes. All the offspring will have brown eyes. So yeah, in that case, all four genotypes have both uppercase B and lowercase b. So they all have both of them have uppercase B and lowercase b, which means they are all bb. But okay, or bb, so but it is the same thing. So since bb is heterozygous, so because it is not of the same gene, is not of the same gene, one is uppercase and the other one is lowercase, so they are heterozygous, assuming if it is, if both is dominant, so it will be homozygous, or if both is recessor, it will be it will be homozygous. But since we have one dominant and we have one recessive, so it will be heterozygous. So since BB is heterozygous and B is dominant, all four genotypes will express the dominant trait, which is brown eyes. As you can see, they all have they all offspring have brown eyes. So we expect the dominant trait, which is brown eyes. So the correct ratio is 1:blue, which is 4:0. So as you can count it, if you count it, there are four. There are brown fours. The brown are four, and while the blue are we don't have any offspring that have blue eyes. So now, or as a percentage, it will be brown 100%. As I said earlier, the square is that is you know 30, that is 25, 25, 25, 25. So 25 plus 25 plus 25 plus 25, so 25 in four places, it will be 100. So brown will be 100%, while blue will be 0%. So in other words, all four offspring will have brown eyes and none will have blue eyes. So I believe we understand the example of how Punnett square are create and how we apply it. So now let's talk about the application of how Punnett square is being applied. Punnett square have many practical uses in various fields, including one in genetics. So it is applied in genetics, that is predicting the probability of inherited traits and genetic disorder. So we use Punnett square to apply in genetics, for instance in maybe a sickle cell child. So it allow us to be able to know if the child inherits the trait of sickle cell or not. So we also applied it in breeding, that is selecting for desired traits in animals and plants. So to know the plant to plant, I mean to know the flower to plant or the animal to rear. So it also applied in breeding. So three, we use Punnett square to apply in forensic science, so to analyze DNA evidence in criminal investigation. So it is also used in forensic science. So four, it is also used in medicine, so to understand genetics risk and counseling patients on sickle cell. In our last class, so we use it. We talk about sickle cell. So to all know if maybe if the mother is SS and the father is AS, so to be able to understand the risk and to be able to counsel them. So we can also use Punnett square to do that. So it's also used in agriculture, so to develop new crop varieties and desirable traits. So to know the crops to plant. So to know the variety of crop to plant, it is also an the desired crop to plant. So we also use Punnett square in the application for agriculture. So the sixth uses of Punnett square. We also use it in evolutionary biology, that is studying population dynamics and evolution. So that is how the application for Punnett squares, how it is used in various fields. So to recap everything that we have covered, to recap everything, so we speak on Mendelian inheritance, that is predicting genotype and phenotype probabilities using Punnett squares. We also talk about understanding dominant and recessive traits. We also use it in applying. Okay, we also talk about how we can apply Mendelian inheritance to real world scenarios such as genetics, agriculture, medicine, forensic science, and so on. So the takeaway keys. So what we should know is that Punnett squares helps predict genotypes and phenotypes probabilities. It also helps in genotype and phenotype okay, that genotype and phenotype are not always the same. So we understand that genotype and phenotype are not always the same. And the third key is that Mendelian inheritance has practical applications in various fields. So now maybe let's do this together. So let's solve this Punnett square together. So this okay, we want to know the probability of red and pink flowers in this diagram. So what will be the probability of red and pink flowers? So the G represents the capital letter G represent the red flower, and the small letter G represents the pink flower. So that is these are heterozygous uh heterozygous how will I put it? Okay,
Sir, which one are you showing? Is it
heterozygous chromosome. Yes, these are okay. The last one.
okay?
Okay, can, can you see it? Yeah, the
Yeah,
last one.
okay. There is no other one.
No, after the application.
Okay, okay. It's still under recap.
Can you see it now?
After the application is
You can see it,
recap.
Abi.
Yes, yes, you
Yeah,
can.
under recap. Yes, it's still under recap.
Okay, sir.
Yeah, yes, it's still under recap. Yeah, under the conclusion. Yes, ma'am. So now we have one dominant G which represents the red flower, and the other which represents the pink flowers. So it will be the result will be capital letter GG in the first column, and the other column will be capital letter G and one small letter G, and in the other row the first column will be one capital letter G and small capital letter G, and in the other column will be two small letter G. So the result will be the result that the probability of red flower is higher than the probability of of uh pink flower. So the probability of red flower will be 75%, while the probability of red flower okay, the probability of red flower will be 75%, while the probability of pink flower will be 25%. So that's concerning the how Punnett squares are applied. So thanks for listening. Is there any question so far concerning maybe Mendelian law of inheritance, how Mendelian law are applied, the Punnett square and the like? So if there is any question.
Okay, yes, sir. You mentioned that I think there were two things under Mendelian of inheritance. The first one is position of gene. The other one is partial something. I can't remember. Please, can you explain segregation of genes, please?
The what?
You explained segregation of genes. I didn't really get.
Okay, the law of segregation.
Yes, yes, yes.
Okay, yeah. That the law of segregation, as I said earlier, it also means separation. That law of segregation also means separation. That is law of separation. So now, okay. So now, I said the law of segregation means that each pair of alleles, that is different form of gene, separate during gamete formation. So it means there will be separation during gamete formation that will later result in each gamete receiving only one allele. So receiving only one allele. And I cite an example
Okay, sir.
when we were
I
talking.
are you, sir? My question is, you said you are receiving one out of those two heterozygous genes and one allele of those two. Is it which one gongon is being sent? Is it the dominant one or the recessive?
Okay, yeah. So the thing is, ah, the dominant, the dominant will be the dominant will be the dominant and the recessive will be separated. You know, the alleles, for instance now, for instance, when we are talking about that law of segregation, I said the alleles will be separated. The alleles will be separated. So it means the dominant and both the dominant and the recessive will be separated. That is, there won't be in there won't be in pairs. So it will be separated according to the diagram I cited earlier. So as you can see that it was separated. The gene was separated. So one for the father and the other one for the mother. So can you get it, sir?
Yeah, I understand it, sir. Thank you.
Okay, okay.
Okay, thanks so much, everybody. I'm not sure if there's any other question for now. So for us to just go back to what we've learned today, I want to ask some questions if we really got everything correctly. Today we explored the Mendel's law of inheritance. Who can tell me or remind me who discovered the law of inheritance? Am I audible? I asked a question. I don't know if you all can hear me. Am I audible?
We can
Okay,
hear you.
I can hear you.
Okay, thank you. I asked a question. Let's go back to what we've learned
Yes.
today that this similarly has taught us to. So, to be sure that we all understood what was taught, I asked the question that who can remind me of the person that discovered the law of inheritance? Can someone kindly remind me of the person that discovered the law of inheritance? Miss Oluwafermi, are you on the call? Well, that's why I'm sharing the screen. Okay.
Mendel, Brother Mendel,
Gregor
Gregor
Mendel.
Mendel.
Thank you. Gregor Mendel in the 19th century. And he's also called the father of genetics. I just want us to remember all these things. And one of the questions I wanted to ask before was part of the answer I gave you when you asked a question that segregation also means what? It also means separation. So anywhere we hear segregation in this particular area, we know that it is separation. And when it was explaining that part, that the blue is what's represented by capital letter B. Can you hear me? Okay. Okay, sorry, my network was bad. So the blue is represented by what? Capital letter B allele. While the red is represented by small letter b allele. I don't know if you remember that part. If you remember, just react to it. Let's see. Okay, good. Thank you so much for that. So the second question is the word assortment. It means what? The word assortment. I think I should go there. The word assortment is what? Can anybody remind us? Don't know where that is anymore.
Okay, the word assortment, selection of various items, selection of various things.
Yeah, it is. Thank you for that. Using another word. Okay, it is what distribution or grouping. Like selection is true, but the word in short is grouping or distribution. So for us to just understand the key point under the law of dependent assortment, the term assortment means distribution or grouping. I want us to read more about that Punnett square very well. I was able to understand in that area that how we all got the black hair and the brown hair, and how we all got our brown eyes. I don't know if because in Nigeria I've not seen anybody that has this kind of eyes, blue eyes. Most of us we have either black or kind of white, brown. I don't know if I'm right, Mr. Justin Lane.
Yes, you are right, ma.
Okay, thank you so much. So, which areas do we use Punnett squares?
Yeah, you are right, ma.
Okay, thank you. So, who can tell me which areas we use Punnett squares in many areas, but who can tell me the key parts like where we use Punnett squares? Okay, let me go to that place. Okay, good. If we check very well, Punnett squares have many practical uses in various fields, including what? Genetics, breeding, and forensic science, medicine, agriculture, evolutionary biology. So Punnett, we use Punnett square in various practical ways in various fields. And I want us to read more about it. So I believe that we all learned a lot today. Hope I'm audible, please.
Yes, we can hear you, ma.
All right, thank you so much.
You can hear you, yes, ma'am.
Okay, thank you so much, everybody. Please give Mr. Discipline a thumbs up if you enjoyed the session tonight. All right, thank you so much, everybody. It was nice having Mr. Discipline here. The session was great, too. If anyone has any
Same here,
question,
ma.
if you have any further question, you can ask on the group. Then we'll continue from there.